You're listening to This Week in Cardiology from theheart.org, Medscape Cardiology. This podcast is intended for healthcare professionals only. Any views expressed are the presenter's own and do not necessarily reflect the views of WebMD or Medscape.
When every decision matters, you need trusted medical intelligence. Medscape AI synthesizes peer-reviewed evidence and Medscape's expert-validated content in seconds, delivering clinically verified answers you can trust. Join over 13 million Medscape members on the world's leading platform for clinicians. Start using Medscape AI on the web or in the Medscape app. Medscape AI, trusted medical intelligence.
Hi everyone, this is John Mandrola from theheart.org, Medscape Cardiology, and this is This Week in Cardiology for May 22nd, 2026. This week, 3 more Digoxin trials, yet another GLP drug on the horizon, vagal nerve stimulation, trial inside baseball, and more on the lipid guidelines. So I want to talk first about low-dose digoxin in heart failure, the Decision trial. And I almost missed this big digoxin trial. At the ESC Heart Failure Congress in Vienna last weekend, Professor Dirk Jan van Veldhuizen from Groningen, Netherlands, and I'm sorry if I mispronounced all that, presented results of the Decision trial of low-dose digoxin in patients with HFrEF. The results were published simultaneously in Nature Medicine about 10 days ago, and I missed them last week, even though I discussed the highly positive DIG-RHD trial of digoxin in patients with rheumatic disease. The thing is, the Decision trial has been anticipated since the 2025 publication of the positive but statistically fragile results of the DIGIT-HF trial. Decision randomized 1,000 patients with symptomatic heart failure and a reduced ejection fraction, or at least ejection fraction less than 50%, to low-dose DIG or placebo with the target serum DIG concentration of 0.5 to 0.9 nanograms per milliliter. The trial was carried out in 43 sites in the Netherlands. The mean age of patients, 73 years, 28% female, and almost a third had atrial fib, which is actually an important point. Mean LVEF was 33 to 35%, and importantly, at baseline, contemporary GDMT was excellent. So the Decision trial was a real test of add-on DIG to the contemporary therapy. The primary endpoint was a composite of total worsening heart failure, that's heart failure hospitalizations or total urgent heart failure visits, or CV death. And that's a little bit different than time to first event. The trial was powered to detect a 22% reduction in the primary endpoint. Now the results. The primary outcome incidences were 3.6% lower in the DIG arm. It was 15.7 versus 19.3 per 100 patient years. Hazard ratio 0.81, confidence intervals sadly 0.61 to 1.07, so the p-value was 0.13, which is above that 0.05 magical threshold. Now, most of this non-significant reduction was due to worsening heart failure events. The heart failure event number was hazard ratio 0.76, but again, confidence intervals above 1, 0.54 to 1.05. CV death was about the same, 5.5 versus 5.8 per 100 patient years. And death from any cause was also similar, 7.7 versus 8.4 per 100 patient years. Also, unlike many heart failure manuscripts, the Decision authors tell us total hospitalizations, thank goodness, which were similar. One of the reasons that a 19% reduction in the primary endpoint did not reach the statistical threshold was a higher than expected dropout rate, potentially due to COVID per the author's discussion. 24% in the DIG arm and 21% in the placebo arm stopped the study drug for reasons other than death. A sensitivity analysis using an as-treated only patients revealed a 34% reduction in the primary outcome. That hazard ratio was 0.66 with tight confidence intervals, but of course, we worry about as-treated analyses as patients who remain on the drug may be different than those who discontinued. Now, treatment-related adverse events were similar. So my comments, technically, The decision trial delivered null results. The absolute risk reduction of 3.6%, NNT of 27, is indeed clinically meaningful but did not reach statistical significance. So, I suppose we would say that if you assume the null hypothesis of no difference, these results are not surprising enough and could therefore be due to chance. Yet, if you take them into the context of the previous trials, that is, the DIG trial and the DIGIT-HF trial, the results are directionally consistent, aren't they? There were not differences in serious adverse events, which is reassuring. Now, the problem the trialist had, I think, was not recruiting enough patients. So technically, you would or could call this a negative trial, but I think that's too harsh a frame. The majority of the 95% confidence interval was to the reduction side. What's more, if I inputted the positive results of the DIGIT-HF trial, albeit with Digoxin, not Digoxin, and I do it into a Bayesian format, which I think is quite appropriate given the similar patient populations, you get something along these lines, and it makes total sense. You get a 99% probability of any benefit of the primary endpoint over placebo. So anything greater than zero effect is highly likely. You get an 82% probability of more than a 10% benefit, but only a 24% probability of a 20% benefit. I sent the numbers to Jay Brophy at McGill University, another expert in Bayes, and he got similar estimates, as did Sanjay Kaul. Strong probability that DIG had a greater than nothing effect, but even using the enthusiastic priors from DIGIT-HF, the probability of more than a 20% relative risk reduction was quite low at 24%. My take, therefore, is that it was a null trial, and we should call it that, but there was no harm, and it was directionally consistent with DIGIT-HF and DIG trials. Okay, so what do we do when we have three trials? We meta-analyze them. So in JAMA, on May 10th, 12 days ago, a meta-analysis of the 3 DIG trials in HFrEF, including DIG, DIGIT-HF, and Decision, and the authors were mostly from the Decision and DIGIT-HF trials. And when they combined the 3 trials, it yielded a sample size of 9,000 patients randomized. The mean age, 64, 22% female, and the composite of CV death or worsening heart failure occurred in 41% in the DIG arms versus 45% in the placebo arm. That hazard ratio is 0.85, confidence interval 0.80 to 0.90, and a highly significant p-value. First worsening heart failure event occurred in 26% of patients in the DIG arm versus 33% in the placebo arm, hazard ratio 0.75, so a 25% reduction, highly statistically significant. CV death was 27% in both arms. All-cause death was also 32 and 33% respectively. The authors did a few sensitivity analyses, and they found that DIG effect was similar regardless of the background therapy. So there was very little background therapy in the old DIG trial, and there was a lot of background therapy in Decision and DIGIT-HF, and the results were similar. They were also similar using a fixed or random effects types of analyses. The authors state that the effect size for risk reduction of the composite primary was about 15%, and it didn't matter the baseline event rate. The baseline event rate was high in the old DIG trial, a little bit lower in the newer trials. The driver of the effect was obviously a reduction in heart failure events and not so much CV death. And regarding the lack of mortality effect, the authors rightly state the following, quote, "The discrepancy between the absence of a reduction in mortality and the reduction of worsening heart failure events associated with digitalis glycosides is not unique and was also observed with the sodium glucose co-transporters and ivabradine and finerenone in the Fine Arts HF study." Okay, the third study I wanted to mention is the Decision Withdrawal study. This is a really clever study. The European Heart Journal has published this super clever report from Decision on what happens at the end of the study with digoxin withdrawal. Now, as background, you recall the very old Radiant study. This was Milton Packer-led Radiance trial where they randomized 178 patients with heart failure and low EF to continue receiving DIG or to take placebo. and they found a 6 times higher rate of worsening heart failure events in the withdrawal group. Well, in Decision, at the end of the trial, 587 patients were on active therapy, 288 on DIG, 299 on placebo, and these patients underwent blinded withdrawal. During the 100 days pre-withdrawal, the incidence of CV death or worsening heart failure events were pretty close together. They were a little lower for DIG, 5.7 for DIG, 6.5 per patient, 100 patient years for placebo. Hazard ratio is 0.88 and confidence interval 0.24 to 3.10. But following withdrawal, the incidence rate increased markedly in patients withdrawn from digoxin but not from placebo. It was 42.8 versus 5.9 events per 100 patient years. That's a highly significant p-value. It was 14 heart failure events versus 2. The relative risk increased 7.37, with confidence intervals going from 1.5 to 35. Now, I know what you're thinking. You're saying, those are small numbers of events, 14 versus 2. The confidence intervals are wide, but consider mechanistically, too. Withdrawal of DIG also led to a statistically significant increase in heart rate, reduction in blood pressure, and rise in NT-proBNP. Now, the authors tell us that not only is this similar to the old Radiant study, but they also remind us of one of the most clever reanalyses ever done, and I want to mention this. Recall that the DIG trial, the old DIG trial, was NIH funded, which means the data eventually becomes available to other authors for further study. Well, 14 years after publication, a group led by Dr. Ali Ahmed and Michael Rich studied discontinuation of digoxin in the DIG trial. They did this by looking at the subgroup of patients who entered the trial while taking digoxin and then randomized to either staying on digoxin or changing to placebo. And so if a patient was on digoxin chronically and they were randomized to placebo, that is a discontinuation. Their papers published in the American Journal of Cardiology, and there were 7,800 participants in the DIG trial. 3,300 received digoxin before randomization. So during the trial, about half of those were randomized to the DIG arm and thus continued to receive their long-term DIG therapy. However, 1,700 patients were randomized to the placebo arm, and thus their long-term digoxin therapy was discontinued during the trial. Serum digoxin concentrations were made at random during the DIG trial, and of the 1,600 patients who continued on DIG, 457 had low levels, that is 0.5 to 0.9, and 340 had high levels after 1 month of therapy. So now the authors had a three-way comparison. They had a discontinued DIG in 1,700 patients. They had a continued DIG with low serum concentrations, 457, and discontinued DIG with high serum concentrations, 340. There were some small differences in baseline characteristics. For instance, those with high serum digoxin concentrations were slightly older and more often female. So the results, they presented two main results, a result for all-cause mortality, and remember that was the primary endpoint of the DIG trial. And then they also present results for heart failure events. So all-cause mortality, listen carefully to this, 38% in the discontinue arm, 32% in the continue arm with low serum concentrations, hazard ratio 0.73, with confidence interval 0.61 to 0.87, but 45% in the continue with high serum DIG concentrations. So that hazard ratio is 1.17 and confidence interval 0.98 to 1.39. Heart failure events, hospitalizations occurred in 40% of patients in the discontinue group, but 29% of those in the low serum DIG concentration, that's hazard ratio 0.61, with confidence intervals going to 0.5 to 0.73. And 34% of those in the high serum digoxin concentration group, 0.78. And again, that was statistically significant as well. So my conclusion, similar to the Decision trial and the Radiance trial, discontinuation of long-term DIG therapy was associated with an increase in mortality compared to the low serum DIG concentration group and worsening heart failure events in both. I would take from the withdrawal data a couple of lessons. One is that if a patient comes to you on digoxin and is doing well, and you are titrating up the GDMT, do not stop the digoxin. Withdrawal does seem like it is associated with harm. The second lesson is that the withdrawal data lends support for the findings of the DIG, DIGIT-HF, and decision data, in my opinion. DIG skeptics may say the effect size of digoxin is modest, the effect is only on heart failure events, not CV death, and the drug requires careful usage and some periodic blood monitoring for levels. And my counter to that is that its effect size is clinically meaningful. The reduction in heart failure is similar to that of SGLT2 inhibitors, and 2 of the 3 trials, Decision and DIGIT-HF, included patients with atrial fibrillation, and there's a lot of AF patients with heart failure. And remember that the beta blocker data with heart failure is very much concentrated in patients with sinus rhythm. The Kocheka meta-analysis in Lancet showed that when you meta-analyze all of the beta blocker heart failure trials, you find that the benefit of beta blockers are only in patients with sinus rhythm. So when we look at the DIG trials, here we have at least 2 trials that include patients with atrial fibrillation, and there's positive effects. Now, the third lesson I would take from this data is more nuanced, and that is that many of our foundational heart failure therapies, ACE inhibitors, carvedilol, MRAs in the RAILS trial, spironolactone, for instance, they were all shown to be effective in patients taking digoxin. When you look at baseline characteristics of those old trials, you'll find that a high percentage of patients were on digoxin. Now, do you think that may have had an effect? For instance, digoxin raises blood pressure. That may have benefited or made it more beneficial for these drugs that lower blood pressure. Digoxin has a positive inotropic effect. That too could have modified the benefits of these foundational therapies. We start foundational therapies in 2026 rarely with DIG on board, but when they were studied back in the day, these patients were on digoxin, and I think that's something to think about. A final conclusion. Think about DIG use in heart failure. If your patient has AF, rather than pushing beta blockers higher, maybe add digoxin. For the patient with soft blood pressure who has limited ability to take good doses of ARNI or beta blocker, consider digoxin. Of course, you always have to use it carefully. It's cleared via the kidney, so adjust the dose and check levels. But DIG, I think, can help many of our patients. Now we have contemporary data that is directionally consistent. I think the main headwind for DIG use is that there will be no shiny, happy pharma reps bringing free bagels in the morning and burritos for lunch. There will be no pictures with banners and cake celebrating the 100th use of some dubious cardiac device. You just have to think about DIG use on your own with no marketing help. All right, next topic is yet another GLP-1 drug announced this week. Lilly released results of the TRIUMPH-1 trial for their new GLP drug called retatrutide. Gosh, I wish they wouldn't make these drugs so hard to pronounce. This drug is actually a triple incretin receptor agonist acting on the GLP-1 receptor, the GIP, which is glucose-dependent insulinotropic polypeptide, and the glucagon receptor. TRIUMPH randomized 2,400 patients with obesity, 1 comorbidity, and no diabetes. The study evaluated multiple doses. Patients on the 12 milligram dose lost an average of 70 pounds, or 28% of their body weight over 80 weeks, with 45% of patients achieving a greater than 30% weight loss. Individuals with a baseline BMI greater than or equal to 35 who participated in a study extension continued to lose weight and achieved up to an average of 85 pounds of weight loss. At the 4 milligram dose, reached with only a single escalation step, participants lost an average of 47 pounds, 19% at 80 weeks, with a lower observed discontinuation rate due to adverse events, at least versus placebo. Now, side effects do seem slightly more common, which makes sense because I think it seems the more potent these drugs are, the more nausea, diarrhea, vomiting, and constipation there will be. It may be that tirzepatide may have a better ratio of efficacy to adverse effects. The company says the TRIUMPH-1 trial will be reported at an upcoming ADA meeting, and surely this drug will gain approval, and yet there will be another GLP-1-like drug for patients with obesity. My friends, I don't think I'm wrong about this. I mean, maybe I am, but given the tremendous rise in obesity in the U.S. and other Western countries, these drugs may end up being more of an advance than statins. Of course, the main focus is on weight loss, but there's also reductions in blood pressure, an improvement in glucose handling, and atherogenic lipids are lower. And of course, we know that semaglutide has been shown to be a disease-modifying drug in patients with established ASCVD. I hold little doubt that they will also have a primary prevention properties in ASCV, and they're likely going to be AFib-preventing drugs as well. Now, one place they're probably not going to be helpful is in patients with HFrEF, as they have a substantial increase in heart rate. All right, I want to tell you a big story in heart failure science regarding vagal nerve stimulation and HFrEF, and this gets into inside baseball, and it makes us think about how medical science is generated. Jack has published results of the ANTHEM HFrEF trial of vagal stimulation. This trial was an FDA-informed pivotal trial examining efficacy of vagal nerve stimulation in patients with HFrEF who remain symptomatic on GDMT. Now, VNS, or vagal nerve stimulation, proceeds via a proprietary system of right cervical vagus stimulation, and from the protocol paper, VNS is delivered using an atraumatic self-sizing lead cuff and specifically designed lead and pulse generator, think pacemaker, to electrically activate the vagus nerve in a manner that produces both afferent and efferent functional effects. The VNS intensity is up-titrated subsequently in the ambulatory setting, and titration targets a small change in beat-to-beat distribution of R-to-R intervals during a 14-second on time of the VNS duty cycle compared with preceding off time. The randomization in ANTHEM was 2-to-1, 532 patients. The primary endpoint was typical endpoint, CV death or heart failure hospitalizations, and the secondary endpoints were also typical, things like quality of life, questionnaires, LVEF. Sadly, after enrolling 532 patients, the sponsor terminated enrollment, and this was unrelated to futility or efficacy. The primary efficacy endpoint was not met. Hazard ratio 0.84, confidence interval 0.62 to 1.12, so the p-value is 0.11, which is above that threshold. The primary safety endpoint was achieved, 97% freedom from procedure or device-related adverse effects, and the actual autonomic engagement persisted long-term. The still blinded investigators sought to extend data reporting while maintaining scientific integrity, and they had pre-specified a single exploratory outcomes symptoms win ratio. And of course, this win ratio, which includes the questionnaire and quality of life issues, was favorable, although it's merely hypothesis generating given the primary endpoint being neutral, and that there's multiplicity and subjective bias and marginal statistical significance. The conclusion of the paper, published in JACC, was that early termination rendered the results uninformative. Now, the controversy comes because the DSMB evaluation of the trial results led to a recommendation to continue enrolling patients. They apparently did not find criteria for futility. Now, here comes the inside baseball part. And I think I have this correct. The sponsors, a company called LivaNova, received a breakthrough device designation from FDA. They also received flexibility to use a Bayesian adaptive design, which would allow consideration of a smaller sample size if evidence of benefit was identified at an interim analysis. And so based on these parameters, the ANTHEM trial included scheduled interim analyses starting at 400 patients, had been randomized, and again after every additional 100 patients to assess the futility of further accrual. Then, boom, in the fall of 2022, The second interim analysis was conducted at the trial's DSMB, found that, quote, the pre-specified conditions for early FDA filing had not been met, and so the trialists wanted to keep going and randomize more patients. Basically, we needed more data. But the sponsor did not like this. Apparently, they wanted early filing. They did their own analysis, and a few months later, after not receiving this early filing criteria, said the trial was done. They were done. So later, as an editorial accompanying the manuscript noted, this decision was financial and motivation, which is like saying you get wet riding a bike in the rain. Of course, the decision was financial. The thing is that 532 patients had been experimented on. And the trialists did the right thing in reporting the underpowered, uncertain results. Their attempts to make more of the data with the win ratio was also laudable, but it's still uncertain data. One idea put forth by the editorialists was that if FDA does give a sponsor special regulatory designation for a breakthrough device, there ought to be also an explicit condition on which trial termination can take place. Here, the sponsor overrode the DSMB solely because they calculated the device was not going to make it, or not going to make enough money. But it might have made a positive result, and we would have learned something. As I've said many times on this podcast, these are the downsides of dependence on so much industry-sponsored science. When the confluence of interest between a profit motive and learning move away from each other even a little bit, industry science sort of fails, doesn't it? I mean, we like industry. They help us. They help make better devices, they iterate, and patient care gets better, and they make more money. But the nanosecond that confluence of interest comes apart, then it's a problem. I think a better way for medical science would be that the government funding was responsible for the final step, these phase 3 trials, an independent body studying the phase 3 studies. That way, a neutral party adjudicates efficacy, and we don't end up with early termination of trials like Anthem, and we don't end up with horribly biased endpoints, such as those that we saw in Option or Champion AF. All right, final topic today is about lipid guidelines. As many of you know, my most recent column on Medscape chronicled 4 major concerns I had with the new lipid guidelines. I worried about A, the discordance between evidence and the class of recommendations, B, changing thresholds for treatment with the prevent equation, C, undue complexity of the guidelines, and finally, conflicts of interest. I was especially put off by the use of CAC, lipoprotein little a, and emphasis on such early and repeated screening. Now, it's had some good attention. The piece has 846 likes, 33 comments, and it's the most emailed piece on Medscape right now, so that's nice. Well, coming soon is a rebuttal from the authors of the guidelines, led by Dr. Richard Blumenthal. Now, I'm not going to comment further on their thoughts. I've read their piece. Rather, I would encourage you to go to the website and read both columns. Read me and read their response and see what you think. Finally, on the matter of lipid treatment, there's also one more good piece to talk about, and that's the JACC editor-in-chief, Harlan Krumholz, has a very provocative editorial on the nocebo effect of statins. He wrote this in his editor's column. He was commenting on a recent commentary where a doctor described a patient with a coronary artery calcium score of 280, an LDL cholesterol of 168, a father who died of a heart attack at 58, and this patient had stopped her statin because she felt foggy and achy. Her decision reinforced by what she had read online. At the same time, she was injecting BPC 157, the synthetic peptide with little evidence of safety or efficacy, into her thigh 3 times a week for a knee injury on the recommendation of a podcast. HMK went on to describe the problem with statin nocebos, and I call it that because it surely is a nocebo effect, and the evidence for this is as strong as it gets. Evidence A, blinded randomized trials of statins find that discontinuation of statin is similar to placebo. That is, if patients don't know what they're on, there's no more adverse effect than a placebo. And finally, Francis Wood and James Howard's glorious Samson N of 1 trial in statin-intolerant patients clearly showed that statin pills cause the same ill health as statin placebos. I will link to the Samson trial because I think it maybe one of the most clever studies in all of modern medicine, bar none. The thing that Dr. Krumholz highlights is that there is so much bad information out there on statins, and it's true. And I don't know what to do about it except to promote and celebrate the good science that underpins the statin's effects. But I also think we need to be humble and graceful about patient values. For instance, I'm now of the age where cardiovascular disease prevention is relevant. I look at my numbers and risk, it's borderline, and when considering a statin, I see a small risk reduction from taking a pill every day for 10 years. That's 3,650 tablets to reduce non-fatal events by a few percent. And different people will feel differently about these things. Maximizers will maximize, minimizers will minimize, and our role as trusted expert advisors is to advise. It's also reasonable to consider in a decision to take 3,000 plus tablets in the next decade the skills of our colleagues in the cath labs. They are truly amazing, and as a treatment for a disease gets better and better, the value in prevention or early detection decreases. Finally, I ask, but I don't know the answer to this question, what role will GLP-1 drugs have? What if a person who's overweight takes a GLP drug and the blood pressure, hemoglobin A1C, triglycerides, and LDL plummet? Now a 10-year risk might go to 6%. Then what? Maybe GLP-1 drugs will reduce the benefit of statins. Let me know what you think. So before I close, I just want to say that next week, This Week in Cardiology will transmit from Nyborg, Denmark. I will be at the Danish Cardiac Society meeting talking about evidence and a value, and I'm looking forward to seeing my Danish colleagues. Remember, if you like this podcast, please take the time to give us a rating, write us a review. That helps others find us. Until next week, this is John Mandrola from the heart.org Medscape Cardiology.
You're listening to This Week in Cardiology from the heart.org Medscape Cardiology. This podcast is intended for healthcare professionals only. Any views expressed are the presenter's own and do not necessarily reflect the views of WebMD or Medscape.